So in this video, I want to talk about the basics of shoots or stems. And we think about stems in trees, and we think about the trunk and the branches, and we think, oh, it's just plumbing, right? It's moving water up from the roots, and it's moving photosynthate down from the leaves. But shoots do a lot more than that, and they're actually really interesting. So let's talk about the different aspects of shoots. So number one, if we look over here on the left diagram, we see an illustration of the ends of a branch, right? The twigs. And a couple of things we can notice is, well, all right, so at the tips of the branches we have what's called the terminal bud, and that's where the next section of shoot will form and grow the next year. And then, of course, on the sides we see we have axillary buds on the lateral parts of the stem. And then you can see the leaf scar. So remember that axillary buds form in the little apex between the leaf and the twig. So you can see this little scar right down here where the leaf fell off the previous year, and then the axillary buds in the axis there. The other thing we can see is we can see the bud scars. So you can see right here, and it looks like a little wrinkly part of the twig, and that's where the bud scales from the previous year's terminal bud would have fallen off. So what you can see is that you can see this year's growth, right? You can look at sort of the thinnest and kind of smoothest with thinnest bark, that part of the twig, that part of the shoot, back to the first bud scale right there, or bud scar. And then you can look for another bud scar down here, and we can see, well, that was last year's growth. And then you can keep counting those. You look for those bud scars, and you can count backwards, and you can see the lengths of twigs that grew each year. And of course, as you go backwards to the older and older sections of the shoot, the bark gets thicker and the twig gets thicker. Now, there's two major kinds of growth in plants, right? So we have primary growth over here on the right, and primary growth is basically the apical growth, the lengthening of shoots and roots, right? So each year shoots get longer and roots get longer as well. But then what makes trees and shrubs different from herbaceous plants is they also have secondary growth. And secondary growth is growth in girth, or growth in the diameter of stems and roots. And this is that woody growth that characterizes trees. Okay, so as I said, we looked at something like this in the last slide, right? So we have the terminal bud, and we have apical, axillary or lateral buds. We can see the leaf scars, the bud scale scars, and so on. This is what it looks like if you look at the main trunk of a tree, right? So again, we can count those nodes backwards. If we cut the tree at near the terminal bud, basically we'll see a little bit of tissue here and maybe one growth ring, right? We'll just have the xylem and phloem formed that year. If we go back 2 years, we can see we'll have the cortex, sort of that's the pith, the tissue from the herbaceous stem, surrounded by the xylem and phloem, and then surrounded by another ring. And as we go backwards, we see more and more years. So the lower on the stem you cut, the more growth rings you're going to see. Okay, and here's some of the anatomy of what those stems look like. So if you're talking about just the primary tissue, right, for the primary growth, the growth that's happening that year, this is what it looks like. Right, so we have our xylem and phloem, and those are usually, these vascular bundles, right? It's not a complete ring yet. You see these little circles, and so the xylem tissue you can barely see on this slide is sort of colored pink, and the phloem is colored blue, and so the xylem is on the inside and the phloem is on the outside. In the middle of that ring of those vascular bundles is what we call a pith. And then we have some other tissue out here called the cortex, and then of course we have the epidermis surrounding the stem to keep it, to prevent water loss. Once we get a fully formed year-old woody stem, we see that those vascular bundles are starting to form more into a ring, and now between the xylem and the phloem, in yellow here you can see this strip, and that's called the vascular cambium, and that's where cell division happens, where we're creating new xylem cells and new phloem cells. When the stem gets a whole year old and we have a completely formed woody stem, we have the vascular cambium is formed, and we have a complete ring now. Those vascular bundles have linked together in a ring, and so you can see the xylem on the inside and the phloem on the outside in blue, and then there's the vascular cambium in yellow. And so that's where cell division is going to happen, and every year you'll get another layer of xylem and another layer of phloem. And then we start to get the bark forming, and so we have what we call the cork cambium, which is in sort of like a light brown over here. And the cork cambium is again another layer of living cells that creates the outer bark, or the periderm, for protection and waterproofness and everything else. And then the phloem, as the phloem matures, it becomes the inner bark, which is over here. Okay, there's two main kinds of shoots. There's long shoots and short shoots. And so long shoots are what we think of when we think of shoots, right? The twigs and the branches and the main stem of the tree. But many trees also have what we call short shoots, and these are these little, very slow growing shoots. And often there's, in fruit trees, we'll see buds attached to there, and we often see flowers and fruit attached. And so this is a short shoot over here of, I believe this is an apple tree. Another type of short shoot in conifers is a fascicle. So that little piece of shoot where the bunches of needles attach in pines. And then also in larch, if you look at a larch branch, off of the twig, we have these little short shoots, and that's where those deciduous needles come out. Okay, so here's a cross-section of a mature tree stem, or it's often called the trunk or the bole. And so we can see our layers here again. So this is fully formed. We have the pith in the middle, and that would have formed right when this tree was, that would have been the primary growth when the tree was very short. We have heartwood and sapwood, which is basically the xylem, which we'll talk about in a minute. But all the growth rings here, this is xylem, right? This is the water-conducting tissue moving water up from the roots. Here's the vascular cambium right here, a very thin layer. It's only maybe a cell or a few cells thick. And what that does is it generates a new cell of xylem and then a new cell of phloem as it divides, xylem and phloem. The phloem forms what we call the inner bark, and the phloem is alive as it moves sugars down from the leaves. And then we'll have, on this diagram, there's no cork cambium, but there's a layer of cork cambium here, and that forms the outer bark, that corky protective outer bark. Here's a microscope slide looking at this under a microscope. And so here we can see this layer of vascular cambium. This is living cells, right? Those cells divide, and they elongate, and they fully form, and then they die. And so when we see the more mature cells, so the cells are older as you go this way and you go this way, right? So the xylem, the wood cells, now these are dead. Once they're fully formed and they're hardened and lignified, they die, and these hollow cells are what conducts the water. Here's the phloem cells here, and those are alive and conducting and moving sugars. But as the growth keeps pushing these outward, the phloem cells get crushed. They get crushed between the new young cells of phloem that are growing out from the cambium and the outer bark here. So the inner bark is made of dead, crushed phloem cells. Okay, well, we talked about heartwood and sapwood. So over time, right, so this is the older rings. The older rings of xylem are in here, and the younger rings of xylem are out here, right, because they're being formed here at the vascular cambium around the circumference of the tree. So over time, the older rings, the older layers of xylem get plugged up with stuff, right? They just don't conduct water quite as well. And also the tree actively moves, it can move substances into the older layers of xylem just to get it out of the way, right? It's kind of like a dumping ground. So what you'll notice is that when you cut down a tree, if you look at the cross section here, we have this darker area called the heartwood, and then the lighter area called the sapwood. And so the sapwood is really the rings that are actually moving water up from the roots. And the heartwood is no longer really doing that function because those cells are plugged up. So really it's there for structural support. So this is the way a xylem cell matures. So first we have the sort of primary growth. The cell basically elongates and grows to full size, and so we have the cell wall out here. And then it sort of it grows inward as that cell wall thickens, and it thickens through several different stages. So we have an S1 and an S2 and an S3 stage. And basically what that means is it grows sort of new layers of lignin. So softer plant tissues, more flexible plant tissues are made of cellulose, and then harder stuff, wood, is made of a compound called lignin. That's a very sort of complex molecule. And so this is what happens at those different phases, right? So the primary cell wall is grown first, and then we have the stages where it grows back in towards the center. That hollow area in the middle is called the lumen. And what's interesting about this is at each of these phases, the S1, the S2, and the S3 phase of growth, we have these polymers called microfibrils. And those microfibrils are oriented at different directions. So the S1 phase, we have sort of a net of them crossing like this. The S2, they're at a different angle like this. And the S3, they're at an angle like this. And so that makes them very structurally strong because the microfibril angles are different at each of those stages. And that's what makes wood so sort of flexible and strong. If you've ever seen carbon fiber, like a carbon fiber part of a bicycle, or they make hockey sticks out of carbon fiber now, you'll notice that those fibers are aligned at different angles. So we have multiple layers and different angles. And that's how we get the same strength in wood. Okay, so here's just another cross section, another diagram of stem anatomy. And again, we can see heartwood and sapwood. We can see where the vascular cambium is right there under the bark, the inner bark made of phloem, and the outer bark made of cork. That's called periderm. And we have the cork cambium and the cork out there. A couple of interesting things is here we can see rays. So rays are cells that move laterally from the interior of the tree out. You do have some vertical rays in the phloem, and those are living cells that can move materials throughout the tree. There's lots of things that you can move. What's not really shown in this diagram, but bark is waterproof and typically sort of airtight. So you have to have ways for gas exchange to get into the stem, right? We have living cells there that need to breathe. And so in all kinds of bark, we have little pores called lenticels. And lenticels are, again, just these little holes that allow for gas exchange. On some trees like cherries and birches, those are really obvious, and we'll see that on the arboretum. But in other trees, it's not so obvious, but they're there. Okay, so let's talk about cork. Let's talk about bark a little bit. There's this one interesting species of tree called cork oak, Quercus suber, and it grows in the Mediterranean regions, mostly Spain, Portugal, a little bit in Italy, and also in North Africa. And so cork oak has been used for centuries to make corks, wine corks, and any other sort of applications we need cork for. It takes a tremendous amount of skill. You can see in the upper left, there's a worker taking cork, peeling the cork, the outer bark, off of the tree. And you have to be really careful that you don't damage some of the inner layers. You don't want to damage the vascular cambium or even the inner bark, because that'll damage the tree. But you can harvest the bark, the outer bark, the cork, off of it. And of course, cork is watertight, and so it's really good for capping bottles. It takes a long time to do this. The trees don't really make good thick cork until they're about 25 years old, and even then the cork is all kind of mashed up, right, because of the phloem being jumbled up against the outer bark. So you have to strip the trees when they're 25 years old. Then you have to wait another 10 before you can start harvesting the cork, and then you can only harvest from every tree every about 10 to 12 years. So it takes a lot of trees, but it is sustainable. These trees can live for 200 years and you can keep going back. You don't have to cut down any trees. And then, of course, you can drill out to take those wine corks, and notice how they're cutting in the sort of tangential or the longitudinal section, because if they drilled the corks out this way, those lenticels would be lined up and the corks would leak. Right? Now, wineries will tell you that it's actually better to use plastic corks or screw caps and things like that, and they'll cite all of this stuff about, oh, well, we don't damage trees or whatever, and that's bullshit. These trees this is perfectly sustainable process. There's cork oak plantations that have been operating for hundreds of years. Wineries have switched to plastic corks and screw caps because it's cheaper. They don't have to pay skilled workers to do this. So any claims that cork is non-sustainable is a lie. Okay, let's talk about xylem. Let's talk about wood. Wood is really important. It does the movement of water up from the roots. So it does the plumbing, the water conduction stuff, and it also provides the structural support for the tree to hold the tree up. So we have basically three main types of wood. So in conifers, we have coniferous wood. And then in hardwoods, we have two different types. We have diffuse porous or ring porous. Okay, so in conifers, what you see here is the cells are quite small. And those cells are called tracheids, and the tracheids do two jobs. They move water up from the roots, and they provide the structural support for the tree. So they have to be kind of small with thick cell walls to be able to do that. In a conifer, we have an early wood band, so the tree is growing this way, and this is 1 year's growth that's shown in the slide here. And so in the early part of the season, those cells are larger cell interiors with thinner cell walls. And in the later part of the year, what we call the late wood, the lumens, the interior of the cell is smaller and the cell walls are a little thicker as we go through the growing season. Now in hardwoods, like in most ways, angiosperms are a little more complicated and a little more sophisticated. So in hardwoods, we have two different types of cells to do those two different jobs. So the big cells here are called vessels, and those are really large diameter cells that can conduct a lot of water. And then the smaller cells here, the darker stuff, are called fibers, and those are there for structural support. So they've divided the job into two. The vessels are much more efficient at conducting water because they're so big. There's a law in physics about the diameter of a pipe. And so if you're a civil engineer, you would study that the diameter of the pipe increases flow by a exponent of 4. So basically, if your diameter is 4 times larger, which many of these are at least 4 times larger, the flow rate is 256 times larger, right? So they're far more efficient at moving water, and then the fibers are there for structural support. In diffuse porous, those vessels are scattered throughout the ring. So in both the early wood and the late wood, in the early growing season and late growing season, we have vessels. Ring porous, on the other hand, basically grow some really, really big vessels at the very beginning of the year, right? Again, the tree is growing this way. And then they grow some smaller ones throughout the year, but not nearly as big and not nearly as many. And then the rest of the ring is mostly fibers. And then we can see here's the ring boundary. And then we can see the next spring, we see those big vessels there. So here's an illustration of conifer wood. Again, we see the tree is growing here. So here's in sort of 3D, here's early wood tracheids, right? Those water conducting cells. They get smaller as we go through the late wood. Then we have the dormant season here, and then big cells again. Those cells line up, and you can see these things. These are called perforation pits. And this is how water can flow from one tracheid to another. They don't stack end to end. The tracheids do sort of end. You can see here this tracheid sort of comes together and seals off. So water flowing up through that would have to flow through these perforation pits into another tracheid to keep flowing upward. You can also see in here, here's the rays, so we can see rays moving through the wood this way. And resin ducts. So here we see a resin duct, and that moves resin or pitch through different parts of the tree that it can use to seal off wounds and things like that. Here's another illustration of conifer wood. So again, the tree is growing this way, and we see early wood, right? Lighter cells because they've got thinner walls and bigger interiors, and then darker late wood, which has thicker cell walls, and then growth stops right here. That's the dormant season, and then we start again in the early wood with large cells. You can see, so these are all tracheids. They're in sort of straight rows. We can see the rays, you can see the streaks coming through here. Those are the rays moving materials laterally through the stem. And then here's a resin duct right here that moves pitch, that sticky stuff in conifers. Again, here's just another illustration. So here our tree is growing this way. We go from early wood to late wood, and we can see those straight rows of tracheids. And the early wood and late wood, of course, is what gives us those wonderful sort of alternating dark and light bands in the cross section of conifer wood. Okay, here's a hardwood, and so again, this is diffuse porous, and we can see those vessels are really large. Now the vessels, unlike the tracheids that don't really stack directly on top of each other, the vessels do. And so we can see basically a long pipe of vessels. And so we have, where they join together, when those cells were alive and they're dead now, we have what's called a perforation plate. And so it's just sort of like the end of the cell, and it's got these big holes in it for water to move through. The shape and arrangement of those holes differs from species to species, but in general, water can flow through. We also have pits that connect between vessels, where vessels join together, so it can move laterally too. And then we can see these much smaller cells. These are the fibers that give that structural strength. And again, we have rays that move material laterally through the stem as well. So here's some ring porous, some ring porous species. So again, vessels, most of those big vessels are right in the early wood. So we can see there, sometimes that early wood is several layers of vessels thick, and then the darker material is fibers, which is the structural support, and then you can see the rays running through here, the rays that move this way. There's many examples of species with ring porous wood. So oak over here. Oak is noted for its huge vessels in the early wood, and then really distinct rays. They have really broad strips of rays, many cells wide, that you can see without any magnification. Chestnut, you can see here. We'll talk about chestnut later in the course. Elm, hackberry, sassafras, black locust, hickories, pecan, which is a kind of hickory, all have ring porous wood. Diffuse porous, we don't have those super huge vessels. The vessels are still bigger than tracheids, and they're scattered throughout the ring. Right, so we can see vessels all throughout. We can see rays. The rings, the annual rings, are harder to see, but you can see them because we have this band of what we call terminal parenchyma. So at the end of the growing season, the trees will typically grow this sort of lighter band, and that's how you can see the rings. Many examples of diffuse porous, so sugar maple, red maple, all your maples are diffuse porous, dogwood, and many other hardwoods. Okay, another thing we need to talk about with wood is reaction wood. And so reaction wood is the trees will grow wood of a different sort of structure, and they will grow sort of wider rings in response to sort of physical movement. So imagine we have a tree growing on a slope here, and the soil kind of moves. We have some movement, and so the tree gets tilted. Trees will adjust their growth so they can get growing vertical again. And so if you look on the sides of the road where there's steep slopes, you'll usually see trees of this shape. And the way they straighten themselves up, hardwoods and conifers do this differently. Hardwoods put thicker rings on the uphill side. And they put a lot, and the concentration of fibers in these rings is also really high. So we have this really strong, springy wood that they put on the uphill side to pull themselves back up. So hardwoods pull up. Conifers do kind of the opposite thing. On the downhill side, they put thicker rings, and those rings have more tracheids in them, and so they push themselves up to brace themselves. Not only do trees do this when they're tilted, but reaction wood is also formed at the branches where the branches meet the stem. So the branches don't droop, they hold themselves up. And the same thing, hardwoods put tension wood on the tops of the branches to hold them up, they pull them up. And conifers put compression wood underneath to push them up. And so tension wood that holds branches, that pulls up, and compression wood that pushes up, those are all called reaction wood. So let's talk about the most important, or one of the most important things that stems do, and that's getting water up the tree. So there's a couple of ways to do this. Number one is root pressure. So there's a few species that can actually push water up from the roots. Notably maples, walnuts, and birches will do this. And the way they do that is in the spring, a few weeks before the trees leaf out. So you don't have any demand for water up high in the canopy because the leaves aren't out and respiring. What they'll do is the cells of the roots and some of the cells of the trunk will start to exude sugars. And so they increase the concentration of sugars in the cells, and that draws water up from the roots up into the trunk as the water follows that osmosis gradient, right, from high concentration of water to low concentration in those sugary cells. And this, of course, is the flow of sap in sugar maples and things that you tap to make maple syrup. But root pressure alone isn't enough to get the water up to the top of the canopy. The tops of very tall trees, especially the tallest trees in the world, are almost 400 feet high, the coastal redwoods of California. And we can't get water up that high with root pressure. We can only get up maybe a few meters. So the main way that we get water to the top of the tree is called tension cohesion theory. So water is really sticky. It sticks to itself because we've got these polar molecules, right, where one side of the water molecule is negatively charged and one side is positively charged, so the molecules will stick to each other. We call that cohesion. And you can basically pull a whole chain of molecules up to the top of the tree. As long as that chain of water is continuous. If you get an air bubble in there, it's not going to work. The water will get too heavy and the column of water will fall. But as long as you have that continuous chain intact, you can move water up. I think the physics of this is calculated that you can the physicists have calculated that you can potentially pull water up maybe 130 meters, which is definitely taller than the tallest trees. So this theory works. The way it works is you have solar energy, right? It's driven by solar power. The energy of the sun is evaporating water out of the stomata. The cells near the stomata then lose their water as it evaporates out, and those cells will pull water from the next wettest cell, and then you get a chain of a moisture gradient going all the way out the stomata. That chain works its way back into a vein in the leaf and into the xylem, and then that xylem will pull water all the way from the roots up the stem, out the branch, and out the stomata. The suction required to do this is immense suction, something like 30 atmospheres of, negative 30 atmospheres is the amount of suction at the top of the tree. And actually that will decrease the diameter. So during the day, when this water is really flowing, the tree diameter will actually shrink. You can measure this with a dendrometer. So a device you put around the circumference of the tree, and it measures the circumference of the tree, or measures the diameter of the tree. And you can see that during the day, so here's the trunk is the green line here, and you can see how the diameter is wider at night and then narrower during the day, and wider at night and narrower during the day as the trunk is growing. And that's because that suction is really immense and actually sucks the tree, it decreases the diameter of the tree. Okay, But we have a problem here with this suction, right? So let's imagine you're sharing a milkshake with somebody, somebody that's close to you, somebody that you love. What's more romantic than sharing a nice milkshake? But let's assume that one person had a shorter straw and the other person had a longer straw. So let's say this woman is drinking from a shorter straw, and let's say this guy's straw is much longer. Who's going to get more milkshake? Well, she is, because it takes less energy, less suction to get the water, to get the milkshake through the shorter straw to its destination than the longer straw, right? So this is a problem for trees, right? Because if we have a tree, the lower branches only have to suck through a shorter straw, and the higher branches have to suck through a longer straw. And furthermore, these branches up here are very important to a tree, right? Because that's where they're getting the most sunlight. That's where they're actively trying to grow taller to get over competitors and so on. So you don't want to sacrifice those tall branches, those high branches, over these low branches. So how do we avoid the low branches from taking all the water at the expense of the higher branches? You can pause the video here and think about that, because it's really quite an interesting puzzle. And on the next slide, I'll show you the answer. Okay, so have you thought about it? Well, here's the answer. Around the base of lower branches, the xylem doesn't just go in these straight patterns, these straight channels right out to the branches. It does in the upper branches for the most part, but older branches, the xylem tends to make these swirly features, and we have a lot of dead-end xylem, where instead of the water going right up through the vessels, the vessel will dead end, and it'll have to go sideways out through pits into other vessels. And so the water has a much longer trip. So you could think about it like this. The upper branches have just a straight straw, and they can just suck that water right up, where the lower branches have to drink through a crazy straw, where the water has to kind of make a long trip. And so that's how trees prevent prioritizing lower branches over taller branches. Okay, now the big thing is trees need to avoid water loss. And so that column of water, for this to work, for transport of water from the roots all the way to the top of the tree, for this to work, you need a continuous column of water, right? So you have to have continuous contact of water all the way through those vessels or all the way through those tracheids. If you get a bubble in there, then you're not going to be able to move water up through those cells anymore, right? So we can get a bubble in there, and that's a process called cavitation. So imagine you have some air-filled cells over here, and our functional water-filled cells over here. That column of water is moving up with no problem. But what can happen is if you have really extreme suction, right? So we have a really big drought and we're sucking really hard and not getting enough water, or if you have damage to those cells, or ice crystals can cause this, or especially in the winter, ice crystals will dissolve, and then those dissolved gases, as the ice thaws, will cause gas bubbles or air bubbles in there. And this is a problem. If this continues, you can get suction of this air bubble. We call that cavitation into another area of the xylem, and we can get basically what we call runaway cavitation, or when you have an air bubble in there that prevents the cells from moving, we call that an embolism. So you can get runaway embolism, and if you have enough of that, and enough cells are damaged, and you have air bubbles throughout, then you can have hydraulic failure and the tree can die. So there's a couple of ways that trees deal with that. There's basically two ways that trees deal with preventing cavitation, and that's there's sort of the safe way, and there's the risky but efficient way. So conifers over here and diffuse porous hardwoods, they have pretty small vessels or tracheids. And so that generally prevents cavitation, because the smaller the diameter of the pipe, the less likely you are to get an air bubble. The water sticks to the sides really well and contains a nice column of water, and then the weight of the chain of molecules of water can just pull the chain of water all the way up through the leaves and out the stem out. If you have big vessels like this ring porous hardwood, this oak, those are really susceptible to cavitation because the big diameter, it's easy to get a bubble in there because you don't have that adhesion of the water to the sides. It's not quite as strong. So, the safe way is to have small vessels. The risky way is to have big vessels. But with oaks, because these vessels are so big, they can be so efficient at moving water up. If you lose them, the next year you can just grow some more, and you're not at a big loss. You don't need a lot of sapwood. You don't need many years, many rings of sapwood. You just need one or two. And then the sort of safety valve that oaks have, or that ring porous trees have, is they still make some other smaller vessels. So let's say you cavitate all your your early wood vessels. You can probably keep limping along with just those smaller vessels for the rest of the year, and then the next year you grow a whole new set of big vessels and you're back to where you started. So there's ways around cavitation, but typically once you get an air bubble in a line of vessels, or in a line of conducting cells, they're pretty much non-functional. You can recharge them by root pressure if you're low enough down. So the spring root pressure can recharge those cavitated vessels. And sometimes you can bring in water, rainwater can come in through the stomata, in through the leaves. But in general, when you have a break in that chain of water, the weight of the water is just too heavy to pull up if you have an air bubble in there. You need that continuous column. So trees work very hard by closing stomata and other sort of drought resistant measures to prevent cavitation.